Hello everybody and welcome back. So, since the last lecture we are discussing about some problems, numerical problems we have talked about, some problems and the solutions of the modules what we have so far covered. That means from starting with the module 1 up to module 3. So, these problems and solutions are in combination of both numerical problems as well as today we will come to the synthetic problems. We will talk about synthesis of some compounds. So, last lecture I had left you with this problem that write the correct ionic form of the peptide, and this is the sequence starting with the N terminal alanine, lysine, arginine and aspartic acid, that is the C terminal. What would be the ionic form of this peptide at pH 7.4? This is important. Of which pH you have to write the structure. So, in this case it is pH 7.4. Now, if you have to write the structure, let us start. So, this is alanine is N terminal, which means it has the amine group. So, I am first writing the skeleton and then we will look at the what are the ions or which ionic form they might exist. Alanine is just CH3 here, and then you have CO, NH. Next is lysine. Lysine is 4 CH2, 1, 2, 3, 4, and then a amine. After that, so this would be CO, NH, sorry, this should be the down the plane, one, two, three, 1, 2, 3, 4. This should be off the plane. Arginine. Arginine has 1, 2, 3 carbon. 1, 2, 3. 1, 2, 3 carbon, and then you have NH, double bond NH, and here is NH2. I guess that is the structure of arginine, you can verify. CO, NH, and there is aspartic acid. Aspartic acid is CH2, OH, and here this is the carboxyl terminal. So, this has to be double bond H. So, this is the raw skeleton without going to any charge formation or without going to any ionic characters. Now, let us see what happens at pH 7.4. So, if you remember the pKa values, or if you look at the table of the pKa values that was given for all the amino acids, you will see a general trend. The trend is for amine groups, be it the NH2 of the amino acid itself or be it the amine of the side chains, the trend is all of them have the pKa value which is more than 8, at least more than 8. Most of them are above 9, but at least they are more than 8. which means that above pK of or above pH of 8 they will be deprotonated. Below that pH or below the pH 8 it will stay as protonated form. So, this form will stay as NH2, NH3 plus will be pure NH2 only at least above pH of 8. Most of the times it is above 9. If you look at the table you will see, but in general at least it is more than 8. So, if you want to get the deprotonated version, that your pH of the solution has to be more than 8. So, this is will come to the pure NH2, but below that pH this will always exist as ionic form NH3 plus protonated form. So, there you go. If you do it at pH 7.4, which is the physiological pH, so this will stay not as NH2, this will be as NH3 plus. And similarly here NH3 plus, so is here NH3 plus. They will all exist as the protonated form or the positive charge form. Okay. Now, what about the other side chains? Alanine does not have acidic or basic side chains, so no problem here. This is lysine, this is arginine, and this is aspartic acid, has a carboxylic acid as the side chain. Now, the other trend is for the carboxylic acids, they have the pKa value of course below 7 and much below 7. At most I think pKa value of the carboxylic acids in the amino acids were close to at least 4. So, below that is the most that is the highest pKa value actually of the carboxylic acids of the amino acids, I guess. So, which means above that pH, above pH 4 they will exist as the deprotonated form. So, this above pH of 4 will exist as COO minus. Below that pH it will be protonated, not 4. I mean, it depends on the amino acids, which amino acid we are talking about. You will see that many of them have the pK of the carboxylic acids which are close to 2, 1.8, 2, something like that. So, in those cases, so above 2 or above 3 itself they will be deprotonated, but the general that at least above pH 4 everything would be deprotonated. So, and we are doing it at pH 7.4. So, here in that pH your acid would stay not as acid, but the anionic form. So, this would be the actual ionic structure of the peptide, that all the amines will be in protonated form, all the carboxylates would be in the carboxylic acids would be in the carboxylate anion form. Of course, now you can see that if you change the pH this will vary. If you reduce the pH to maybe 2, then this will all change. If you increase the pH up to 10 or 11, maybe they will also you will also see different kinds of structures possible. Now, you have to deduce, you have to practice what will be the forms at different pHs. So, likewise, so far I have shown you few problems, numericals and as well as based on some structures. So, you can frame your own problems which is closer to this area and then can try to solve this. So, I am just giving you few examples which can have many variations. Okay. So, today we will talk about some synthetic problems. And we will start with a target molecule. NH2, this is cytosine, and here selenium methyl. OH and here ODMT. This is your target molecule. Question is, how can you synthesize this molecule? The change is, for the ribose sugar you have a OH here. In this case we have a SEME, selenium methyl selenide, selenium group basically. How can you synthesize this compound? You can start with your favorite molecule or your molecule of choice. You can choose your own molecule where you want to start from. So, this molecule was synthesized. The reason is there are plenty of variations like this, like SME is there, amines are there, many types of variations that have been synthesized, and they show different kinds of properties actually. So, this particular molecule or this particular kind was synthesized to improve the crystallization property of DNA. If you synthesize the DNA out of this, what was seen that presence of selenium that actually increases the chance of getting good crystals. Usually the crystallization of DNA is very hard actually, and it's a most of the times it fails. It's quite hard to get a proper crystals of DNA if you want to study the structural pattern of a DNA. So, introduction of a selenium moiety was seen that it will increase the tendency to form the crystals. So, there you go, this is the target molecule. Obviously, only one thing you are doing is replacing the hydroxyl group with a selenomethyl, and both of these are down the plane. So, what they have done is they have started with this molecule, writing cytosine, and since you need the selenomethyl in the down the plane, they have started with the OH which was up the plane, because then it makes it easier, and then the as usual. I think that is only the clever part probably, because the rest of these, the others you can easily do. So, first thing you have to do, as we have talked, that if you have a nucleobase, so and in order to make sure that they do not participate in any reactions that you intend to do on the sugars, then you protect the functional groups or the reactive functional groups of the bases. In this case, you have a free amine in the cytosine, and therefore, you definitely need a suitable protection of the amine group. So, you can do it with the acetyl protection, or they do it with this kind of protecting group. If you use this, of course, you get the elimination nucleophilic substitution of the chloride. And what you have is here NaHCO isopropyl amide linkage basically, which is less reactive and it will be stable. I am sorry, I made a mistake in the target. This should be the double bond here. So, this is the double bond. This is the target with the double bond. So, that is the first step. Second step is that you need to keep this OH as it is and you need to protect the other two, so that they do not interfere in your future nucleophilic substitution reaction. So, and again you can see this is 1,3 diol. So, you can protect it by the silyl group, that is TIP D SiCl2, and of course, you need the suitable solvents and all this. If you do that, so the structure of this was, I think I had mentioned once, Cl. This is TIPD, TIPDSCl actually. So, this is the structure of it. So, if you do that, then what you get is, so I am writing this as C prime because this will be all along there, the protection. This is not going to react because mostly 1,3 will react, Si, O, Si, and this. This would be the protection group. Now, once you have protected the 1 and 3, now you can do reactions on this center. Any nucleophilic substitution you do will happen here, because the cytosine will not react, this is not going to react, only position is left is the C2 prime. You have the OH group and that you want to replace, and we know that ether bond or the alcohol bonds are CO bonds basically, right, COH. This bond, CO bond, is quite strong, it does not get eliminated easily. If you remember, OH or the OR, ether bond or the alcohol bonds, are very hard to break, because the OH group or the OR groups, they are not good leaving groups, they are very bad leaving groups. And that is why you need to use usually very strong acid to, if you want to make it protonated, and then only it can be eliminated. So, either you have to use a very strong acid which will make problems here, or you have to find other ways to make this OH a better leaving group, and that is done, it is done here actually, that this OH was made into a better leaving group by OTF. So, if you use CF3 SO2Cl, this is in short, this is called O triplet, OTF triplet, because CF3 has electron withdrawing capacity. Here also they are electron withdrawing in nature, So, and you do the protection first here, and once you do that, so OTF, it is a very good leaving group and very widely used in organic chemistry. And here the other two are there. So, this becomes quite a good leaving group, because you see, if you write O triplet is this. So, if this is eliminated, this comes, this goes there, and CF3 also drags the electron density. So, this overall, this will pull the electron density. So, bonded electron with a carbon, for example, would be towards this, and it can leave easily with a nucleophilic, proper nucleophilic substitutions, even if you do not have a very strong nucleophile, that will react. So, this on treatment with now MEAC, AC, ME can be, so this effectively MEAC minus is the nucleophile. So, if this is the nucleophile, that will attack this center, eliminating the triplet. And of course, since the triplet is down the plane, since this is a, since the OTF is up the plane, the nucleophile has to attack from the down the plane. It is a pure SN2 reaction. So, therefore, your ACME comes down the plane as your target is. Now, all you have to do is to cleave this protection, which you can do with a fluoride ion source, as I have talked. Usually use TBAF. You can find the structure of TBAF. That is the fluoride compound. It is ammonium fluoride, basically tetrabutyl ammonium fluoride, F minus. This has ionic form which has a fluoride minus. So, little bit fluoride that will deprotect this, and will bring back C prime OH, bring back this OH. Then you do the DMT protection, DMT chloride. You need pyridine. Pyridine is a solvent that is usually used for DMT protection, because the DMT protection is very sensitive chemistry. Presence of slight amount of acid or slight amount of water will make this will kill this reaction. So, dry pyridine is usually used. ACME, OH, and you have O DMT. DMT chloride, because DMT has a larger size, that only reacts at the 5 prime position. I think I have shown the structure of DMT before. So, DMT is O-methoxy, two benzene rings will have para O-methoxy groups and one benzene ring as it is benzene. So, this is your DMT chloride. Dimethoxy trityl Cl is chloride. This is it. Because of this large size, it goes actually here only. And then if you want to deprotect this, if you want to go for the DNA synthesis, of course, you will keep this on as it is. But if you want to deprotect this, you can simply use the ammonia, ethanol, or other suitable deprotecting agents that will bring back your C. empty. So, here is your target compound. Now, I would like to give you a brief introduction to a chemistry which is known as, I think maybe many of you are aware of it already, which is known as click chemistry. Click chemistry became very popular because of its applications in chemistry and chemical biology. So, and for that this was developed by Barry Sharpless, K.B. Sharpless, for which he has received the Nobel Prize, because it's a vast array of applications in especially in biology as well as in chemistry, of course. It's basically a coupling reaction, metal coupling reactions that you have seen in many name reactions. They are involved, metal coupling reactions are involved in those. So, here this also is a kind of coupling reactions between an azide. If you have a N3 group azide plus if you have an alkyne, then in presence of copper 1 catalyst, they will be coupled together and they form a 5-membered ring. So, 5-membered triazole ring. Covalent links, and that chemistry is known as the click chemistry. So, it is a reaction between azide plus alkyne in copper 1 using copper 1 catalyst gives you triazole ring, which is in this case, if you do, if you call this Rn3 and if you take this as R prime, this is R prime and this is R, then your product would be here is R, double bond this carbon, here is one N, that is your R prime, here is another N, double bond. Yeah, this would be your, this is the compound, triazole compound. So, this R prime can be anything in this direction, this R can be anything in the other direction, and you can ligate them together, you can join them together by this triazole ring formations. Very easy chemistry, doable, and this can all happen at room temperature, which makes it really a good way for its applications in biology, because you cannot use higher temperature there. So, in this case we will see an example of doing click chemistry for DNA. So, if you have in general a B1 base, if you do a N3 for example here, and you have a DNA where the last base is modified with N3, and you have another DNA B2, let us say this is this is another DNA, the other part of it's one kind of DNA, this is other DNA, DNA 1 and DNA 2. And here if you have 5 prime 1 2 one, then triple bond there. Copper 1, room temperature. Then what will be the product? Of course, product is simple, as you have seen, just that this azide will couple with this alkyne, give you the 5-member ring. So, you will basically connect in this way, you will basically connect the one DNA with the other DNA chemically. This is your one DNA, and what you will have is in another in here, this should be double bond, this should be double bond, here CH2, something like this. Here the angle is not proper, but you can make it better. This is what you will get. Instead of usual, now if you look, the difference between the normal DNA with this molecule is that this is also a DNA, only difference is here the backbone skeleton. Normal DNA has a phosphate bond, phosphate group, here you have a triazyl moiety in this case without a phosphate. This kind of chemistry in biological terms is known as chemical ligation, and that is why this is famous for, because you can you have seen how to ligate two DNA strands using the enzyme, right, DNA ligase. So, in this case you can ligate two DNA using a chemical method. That is why this is known as chemical ligations, and it has found really a lot of applications in biology. If you want to connect one gene with the other, or one modified DNA that you want to use it as a probe with a target DNA, which you can again modify, then you can use the method of chemical ligation in situ to get a or to obtain a variation or to obtain a longer version of the DNA for various of course depending upon your aim and your applications. Similarly, you want to, for example, make a covalent bond between a protein and substrate, which can be a drug or which can be any other biomolecule, can be a DNA, can be the small molecules, for example, vitamins, antibiotics, anything. So, in those cases what you can also do, this has been applied really well there. So, your proteins contains many side chains that has amino groups NH2, which you can easily convert into the azide N3, and then if the other molecule which you want to connect it to, for example, the antibiotic or for example, the other substrate, for example, the drug, if you modify that with an alkyne, then you can easily connect them together and study the protein functions or what the protein actually does in the living cells. So, that is called the chemical ligations. And of course, the other other difference is that you have now, when the backbone becomes a triazyl ring, this is hugely rich in pi electrons, also in heteroatoms. So, this is a planar system. Phosphate is not a planar system, this becomes a planar system. So, this will give you a different orientation of your DNA, and also it will increase the pi stacking interactions as well. So, they are used as a modified versions of DNA as well. So, all you need to do is convert the normal functional groups such as OH here into this. Similarly, here there are many ways to do that. So, the mechanism of this reaction you can find out from book. Click chemistry is quite popular. I will show you here briefly. Now, let us start with a general mechanism. If you have alkyne or C triple bond CH, then the first thing is this binds with a copper. So, this is copper 1. So, I will not write copper 1 again, I will just simply write copper, which means copper 1. So, copper is coming in here, first bind with the pi electron, then comes in here, it eliminates a proton and takes the place of the proton. So, this becomes a copper. Now, another copper comes in, forms a complex here that drags the electron density, and here comes your R prime N3 azide. Now, N3 can be written as this. So, this is neutral, this becomes plus, this becomes minus. So, that is what is your azide is. N3 always exists in, of course, in the resonating structures. You can write other resonating structures as well, but this is one of the form. Now, if this happens, then what will go? I will rewrite this into proper form. You have the copper and then R prime in N R prime in triple bond N. So, this is plus, this is your minus. Now, you have this copper here, that makes a complexation, this makes a complexation. Now, copper is electropositive, so it wants to push the electron density towards this, which increases the electron density on this carbon, that can act as a nucleophile. This will be coming there. So, what you are getting now is a double bond or in R prime with copper and here with the copper, this, and they have, this is originally Sharpless has shown that this nitrogen forms a complexation in this kind of thing, this dotted, but nevertheless this is the raw skeleton that comes here. So, next is of course the elimination of one copper, and then you have here this copper was covalently bonded, you have the double bond, here is R and there is this nitrogen here. Now, it is a double bond and this is nitrogen, with the R prime. Yes, this is what you have. Now, this copper has to be eliminated with replaced by the H. So, this is what it will happen, that H plus, the H plus that you have released here will come and copper 1 would be released, just the reverse of this, and that will give, I am writing it here, that will give you here the H, the central compound, and this copper is eliminated, which can come back to this cycle. So, the cycle can continue. That is why copper 1 acts as the catalyst. So, this is what the proposed mechanism of the reaction, Sharpless reaction. Now, when you are using practically, when you are using the copper 1 as the catalyst, you do not have to use really copper 1 as the key compound. You can use also the copper 2 like CuSO4, which is copper 2, or any other salt, because they are cheaper. And if you want to convert it into, you have to convert it into copper 1, because that is the active catalyst. So, you have to use some, they use ascorbate or ascorbic acid. So, that will reduce this into copper 1. So, this is what is the reaction of the click chemistry. So, I am giving you this, I am talking about the click chemistry just to make you familiar with you, that in future if you are thinking of making any covalent bonds, if you are thinking of connecting to conjugating different kinds of biomolecules, then this is one of the quite popular method to use. Okay. So, now we will move on, and I have talked about the azide. Na? So, now if this is your thymine, this is your thymine, NH, N, this is the sugar, N3 here, and a OH there. So, azide basically. How can you synthesize this molecule? You can start again with the molecule of your choice. So, this molecule is actually a famous drug, famous medicine. This is called AZT. This is one of the first HIV agents, HIV AIDS agent. And very popular medicine, actually very popular drug. So, we have to synthesize the AZT molecule. You can find out the full form of AZT. Shall I tell you? You find it out. You find it out. It is a very famous molecule. So, here they originally they have synthesized it starting with the sugar, but the key material, I mean, from where it actually starts is this. This actually comes from the sugar this is derived from this sugar. Which is D-mannitol. Multi-steps reactions, and it comes down to this. So, from here, if you look from here, how do you get into the compound. So, first thing that you need is the protection over here, which you can do by DMT or you can do by silylations. So, if you use you can use it by silylation also, TB DMS chloride. If you use that, then you can get O TB DMS. You can do it by DMT as well. DMT is little bit fragile. So, if you want to do multiple reactions after DMT, it is better to avoid DMT at the beginning. Now, you have to bring in some functional group here. You can do that, or you have to bring in your nucleobase in this position. So, they have first attached it with the nucleobase here. So, if you want to attach the nucleobase, so the first thing you have to do is to reduce this OH into the alcohol, into the OH. TBDMS. Now, for this reduction, you have to use a reducing agent. Usual reducing agent is sodium borohydride, lithium aluminum hydride and all this, but of course, you know since this is only a single carbon reduction, you want to avoid the multiple H minus, the reagents which has multiple H minus for reductions. So, you use diol here. Diol is a single hydrogen or single hydride transferring agent. Basically, it allows only a single hydride transfer. The structure is, it has a single one hydride, this diisopropyl lithium aluminum hydride basically. So, it has only one H. So, that so it will not affect the other part of the reactions. So, you have OH, which you can convert it into, to do the acetylation, make a better leaving group, little bit better leaving group at least, OAC, OTB, DMS, and from here you can make it further this a better leaving group by reacting it with a triplet OTF. Now, if you directly do this chemistry, okay, I will go to the next page better. Starting here, OAC, I am just writing P for protection. You need a thymine, so thymine, if you start with thymine, and thymine, if you treat it with TMS, because we need we have talked about this when we have done the nucleotide synthesis, that the nucleophilicity here of this carbon is not very good reactive, all the sugars are not very that much reactive. So, you have to increase the nucleophilicity of this of your base basically. So, and we can do that by converting it into the enolate, right. So, you can use TMS or triplet or TMS chloride, both will do, and this will give you the enolate OSIMe3 NO SiMe3, and this basically increases the nucleophilicity here. How? Here or there? you have the OTF minus that was released here. So, this will come immediately, this will go, and that is how your nucleophilicity of through this nitrogen is increased. So, this will react here, eliminating the OH, OAC, and you can have, I am now writing straight T, OP, ready. Now, this is reacted with LiN3 nitrite along with the solvents. So, azide you have T, and then this will include a N3 over here, OP, it can go either there also. So, it can have, you can have a mixture of them, and then a fluoride agent TBAF, that is the fluoride reagent, and get this back. So, one way. Other way is that I will do it quickly. OP you had, first you can do with the LiN3. This is even better actually. I will write it here, better procedure. So, because of this conjugation, your nucleophilic substitutions will probably mainly happen in this case. So, you have this, and your N3 will come here selectively, almost selectively. This is better, this is for me, this is better. And then you can do the reduction and other way. Diol reduction followed by 1 followed by acetic anhydride 2, that will lead you to N3, OP and OAC, and then from there you can add the thymine over there. This is a better way. And lastly, the compounds that we are doing here, that compounds that I am writing the structures of, are all important kinds of compounds which are either biologically found or they are active, they have properties in biological systems. Let us say ODMT, and here is amine. So, this is your usually your cysteine, is not it? Now, cysteine, if you have a aldehyde group here, this is called, from here it will come, this is actually the CH2OH, CH2OH, CH2OH. This derivative of cysteine, this is actually found as a nucleobase in many brain cells, and this has been invented quite recently, I think 6-7 years back, was found a different nucleobase, not in present in all genome, but present in certain kinds of genes or cells, brain cells, or the genes obtained in the brain cells contain a certain quantity of this variation, and this is known as the 6th base pairs, 6th nucleobase. So, ATGC and uracil, these are the 5 nucleobases present in the cells, living cells, and this is the 6th nucleobase that is sometimes present. So, how to synthesize this molecule? I will just show briefly. If you want to make this molecule in large quantity in laboratory, which this amine you have to protect with NHP with a suitable protection group, you can use that, we have done in, and here you start with the iodo compound. From here, using a metal coupling reaction, that is carbonyl insertion basically, phosphine and the other reagents, solvents, these are the key ingredient. CO is the key ingredient in here. See, insertion reaction basically. NHP, then you will have a CO inside, and eventually iodine would be replaced and you will have the H. So, it will come as an aldehyde. I forgot to do one thing. When you start this, you have to start with the protection. So, if you start with the sugar, then you can do the protection. Either you can do TMS, or as we have seen, you can use also the bridged one, that TIPDSCl, TBDMS chloride with the disilylation, which goes to 1 and 3 positions, that also you can do, or if you treat it with TMS, both will be protected. So, here TMS for example, here TMS, and then you have the insertion. This you can reduce with a sodium borohydride or maybe a diol also. Sodium borohydride is fine here. Followed by opening of the, so if this is 1, your second would be your TBAF, NHP, and this would be reduced into the OH, because that is why the reducing agent is 4, and then you can convert it into the DMT. Okay. So, this is a naturally occurring, of course, nucleobase, which you can find a synthetic route to make more amount of in your laboratory. So, these are some of the synthetic problems that I intended to solve and to show that you can play around. So, you can find again, if you have a target molecule, you can figure out in and around of what we have taught, that how to synthesize those kinds of molecules, especially the variations in the nucleic acids. Okay. Thank you.